see these chytrids that have infected this. So the reason I bring those two up is that I've been looking at chytridiomycosis in Colorado. So we have boreal toads that are endangered, and the chytrid fungus is kind of wiping those guys out. But how the chytrid is getting around is not well understood or well known. It's moving very fast through all these kind of habitats. And I was talking to a colleague, and she collected some pond water and left it on her desk for like a week or two, and then threw it under the scope and saw chytrids attacking the pollen that had landed on the surface of the water. So I think that there's a ton of chytrids, and our understanding of how they move is in its infancy. And then also, they're evolving. So we have this new chytrid fungus that infects salamanders, and it was just described in 2015. On the left-hand side is a graph where it's already been found in new places in Europe, and then on the right is where they predict it to start to be problematic in the States. So we're going to see this new chytrid fungus coming into salamander populations within the US. So zygomycota in this group is our black bread molds. So I'll just talk about one group in here, ergot. Maybe you've heard of ergot. It makes lysergic acid. Some folks take that recreationally. And this crazy little fungus, what it does is it infects the flowers of grasses, and in particular wheat and rye. And it has this crazy life cycle, can reinfect the inflorescence. But it also has, I want to bring your attention to the sclerotia. Again, this is, in that talk this morning, Alex was looking at the different biochemical properties and showed that there's different biochemical properties of the sclerotia compared to the sporocarp. And it makes sense because they're doing different stuff. But if you look over here, these grasses, they're rye, they're wheat, those sorts of things. So they make bread out of those things. So ergot, besides making lysergic acid, also causes ergotism. It's a vasoconstrictor, so it dilates the blood vessels and causes all kinds of issues. And there's many, many, many theories. One of the theories of the Salem witch trials, the thing that was causing the people to act like witches, was that they were eating wheat infected with this ergot. So if you have a chance to watch Fantastic Fungi, Suzanne talks about it. There's incredible visuals of them walking on the forest floor and the mycelia sparkling out. So when we think about how this happens, how fungi are using these networks, we think of this hyphal strand, this fungal strand, and all this stuff is moving through these fungal hyphae that are underground. Again, they're on the fringe because we can't really see them. If we start to look inside those hyphal strands, there's all different ways that the fungi are allowing information through their hyphal networks. So this is an example of fringe to me, because we know that gymnosperms and angiosperms are ectomycorrhizal with the types of mushrooms that actually produce sporocarps. Organisms they're attached to, and they're generally characteristic of pathogens or parasitic fungi. So just want to show you this quick video. We talk a lot about hyphae and mycelia, and they're incredibly dynamic. They can respond to stimuli. So if we look at this video, this guy is a nematode. It's a roundworm. This is another very understudied group. There's a ton of diversity. But what we're seeing is this nematode is jabbing into these hyphal networks. And the fungi that we're looking at is this Kopenopsis. So you guys might be familiar with that group, Kopenopsis. So what they did was they looked at the responses of the hyphae when they got jabbed by the nematode. And this is fluorescent microscopy of the protein responses. And what this is showing you is that there's responses differentially. So in one direction, the fungi are producing toxins to say, eat it, nematode. And then they're moving their proteins in another direction to get away from their point of infection. So if you saw Alex's talk earlier, he looked at the biochemistry of hyphae mycelium versus the sporocarps, and noticed that there's a ton of difference in those biochemical properties. And that makes sense because the function is much different. What the mushroom is doing is way different than what the mycelial or hyphal networks are doing. And what this is showing is that it's dynamic. You can change what those things are doing. So, because mushrooms, hyphae, are like, you're not gonna get me, nematode, they've developed these structures where they can actually digest nematodes. So these hyphal strands are sticking to this nematode, and like I said, we exude enzymes. These enzymes are gonna shoot into this nematode and digest that bugger, because they're not gonna put up with getting attacked. No empathy for the nematode? It gets cool. So somebody just sent me this a month or so ago. Hyphae can heal themselves. So what we're seeing is the cytoplasmic streaming of this hyphal strand. It's shooting out all its cellular contents, right? And it's stopping it. It's basically making a scab, just like we make a scab. So they did a lot of work. They knocked out the genes that they thought perform this, and they showed that if you knock those genes out, it can't heal itself. So hyphae are incredibly amazing, and what do they have to do with a fringe? Well, hyphae are moving out into the environment, so they're the fringe, right? They're colonizing the outside. They're looking for more food. They're going into different areas. We also talked about this a little bit earlier today about fungal evolution. So this is a geologic time scale. This is very common information that you're going to get in a textbook. And so what they're showing is, in most textbooks, what we're teaching everybody is that fungi showed up about 450 million years ago. They're saying that it corresponded with a colonization of land. Well, fungi started diversing at that time, but they're much, much older. All right, so our most recent ideas, we've got at least 1 billion-year-old fungi. So the reason that we don't have a good handle of when fungi originally evolved is that they don't fossilize well. We don't see them in the fossil record. So we have to look in things like amber, which makes it really difficult to get information out of those organisms. You've probably seen 100 of these, so I just wanted to go over real quick what we're looking at and how we interpret a phylogenetic tree. So a phylogenetic tree is just the evolutionary relationship of a group of organisms. The tips are taxa. And conventionally, we always had kingdom, phyla, class, order, that sort of delineation. So that taxon could be a species, it could be a genus, it could be a family, etc. So if we look at these trees, we see these branches, and we come back to where the branches meet, and we're going to call that a node. And we can interpret that as these two taxa shared a common ancestor at some point in their evolutionary history. So I just wanted to mention that to remind you how different types that we can talk about. Commensal is where one partner benefits and the other partner is neither helped nor harmed. Parasitic is where one part. So it's a literal highway. It's enabling other microorganisms to move through this medium. And if you see the outside edge of those hyphal strands, you kind of see a shadow there. So there's a sheath around it, and all the hyphal strands have some sort of different type of sheath. But the point for this one is that it creates an aquatic medium. So you see those things that are cruising through, they're bacteria. They're modal. They need some sort of aquatic environment to move through the ecosystem. So those fungal strands enable those bacteria to move through. And as Lauren just mentioned, bacteria are part of this whole mycoremediation process. So if we can harvest or garner all that system, we can really do some cool things when we think about mycelial networks. Hyphae are also super cool because there's tons of modifications. So they can have different functional implications. So hyphae can be modified for dispersal, for dormancy, for protection, or different types of symbioses. And we know this in plants, right? These plant modifications are very common to us. We can think about these like a tendril for climbing, or these pitchers for digesting insects. So just like plant parts have modifications, so do fungal hyphae. So a couple of them that I'll talk about real quick are rhizomorphs, sclerotia, and hostoria. You guys know the honey mushroom, armillaria. These guys make these rhizomorphs. So they're these thick, this is a rhizomorph on a piece of wood, and basically these rhizomorphs are super strong. They've pushed all the bark off, and they can cruise through because they have this super hard outer layer that's melanized. We know melanin protects from UV radiation, so it protects this strand, and if you were to cut that open, inside there'd be hyphal strands inside. So it enables for quick dispersal, so they don't have to complete the life cycle. They can go into new areas, and it gives it a competitive edge because they're physically stronger. So, and this is just a picture of our armillaria malia, the honey mushroom.